Hi, my name is Thomas. I'm going to assume that you guys can all hear me unless Suzanne sends me a message in the chat. So I'm a postdoc in Nicola's lab in Jülich. And actually, let me share my screen before I do anything else. There we go. And I'm going to be talking today about segmenting brain images. Before getting started, I'll direct you to the GitHub that we're going to be using. If you want to follow along with the coding and all that, you can download this file here. Or in Google Colab, you should be able, so if you go to collab.research.google.com or just search Google Colab in search engine. If you go to File, New Notebook, File, Upload Notebook, and then go to GitHub. I can ignore this.
Thomas, could you paste the link to the chat?
Oh, yeah, sorry, I thought that had already been done. There we go. It should be in the chat.
Perfect. Thank you.
Okay, so where was I? Okay, so here you can just copy and paste the link, I think, like this. No, you have to actually copy and paste the link for the Jupyter notebook. There. And then it should open, and then you'll have all of the code that we'll be running today. The instructions are on the GitHub in case you didn't follow that just now. Okay, so I don't know if I go to full screen, if I'll lose the window. That's okay, I'll just leave it like this. So right, today I'm going to be talking about techniques for segmenting the brain or segmenting brain images, starting with very simple techniques like thresholding and then going all the way to neural networks. We only have an hour, so it'll be a very surface overview, but it should still hopefully be useful. I think maybe unlike some of the other talks, this is much more low-level programs that are that you can use in Python. So it's not an overall package, but it's a bunch of different sort of ideas that you can use for your own work if that's useful. And so to make this a bit more interactive, I'll do some short presentations on each of the main topics, and then we'll do some hands-on coding together for 5 to 15 minutes or so. Oh, wait, we see all the other windows you've opened. Okay, is that, then I can't see the chat. Okay, so I'm gonna assume that's good. If not, send me a message. So to start with, we'll have a brief introduction on what segmenting is. And then we'll go over 4 main techniques. So starting with thresholding, then clustering with k-means, watershed segmentation, and then finally neural networks. Neural networks are definitely the most complicated, so I'll try to spend most of our time on that. So to start with, what is image segmentation? So it's very simple. It's basically when you assign pixels from an image to a discrete set of classes. So for example, in the image here, we have a whole brain segmentation from an MRI, presumably. And so you set, for each pixel, you identify which anatomic structure it belongs to. What we're going to be working on today is much simpler. It's just binary tissue classification. So an example would be below, where you see the pen image on the left, and then a classification of whether or not something is brain tissue. So there's lots of different applications for this sort of these sorts of algorithms. One is very simply, if you want to quantify something in your image and you don't want to take the overall intensity value of the image, you have to subdivide it into different regions, subdivide it into meaningful regions, and so segmentation lets you do that. Semantic segmentation also lets you subdivide the image into sort of meaningful units like anatomic regions. It can also be helpful for image registration. So if you have a good segmentation of your image, it can help, you can use it as a mask for when you align two images together. And then just very broadly, it's useful for lots of other downstream image processing. Like with all forms of image processing, there are notable challenges. And these challenges are basically the same as you'd find in other domains of image processing. First, there's just the variability in the images that you may acquire. So even within a given data set, you might have an algorithm, let's say a thresholding algorithm that works well for some of your images, but not for others. Noise can also be a big problem, although maybe a bit less so if we're working with very high resolution postmortem data. It's probably a bigger challenge in PET and MRI. And then finally, there can be a lot of artifacts in your images, which can screw things up. In particular, so here I have some examples of images that I've worked with where you have sort of like these black like pens over the images or the frames. And all of this stuff can make the task of segmenting the image much harder. Okay, so first technique is called Otsu thresholding. This is a very sort of popular and widely used technique, and it's actually quite simple. It's just an algorithm that sort of finds an optimal threshold value such that any pixels that are below the threshold are 0, and everything that is above the threshold is 1. So it basically separates background or 0 and foreground 1. And the way it does this is it finds the threshold that minimizes the interclass variance between the background and the foreground. This is, I think, mathematically the same as maximizing the interclass variance. You don't really need to know that except to understand the little GIF that's on the right here, where you see that as the threshold is moved through the histogram of the image, you see the interclass variance. And so the optimal sort of separation between background and foreground occurs at about like 90 or something like that. So this algorithm has some advantages, notably that it's quite robust. Like if you add some noise to the image, it's not going to stop working. And it's very fast, which can be very important. It does have significant limitations, though. One is that it obviously only separates into 2 classes, background and foreground, which for our purposes today is fine, but maybe for your own applications is insufficient. Maybe a more problematic limitation is that it assumes a bimodal distribution in your image intensities, in your pixel intensities. And so if you don't have that, then it may not work quite so well. Another problem, which I think is quite common with lots of simpler thresholding, simple segmentation algorithms, is that it ignores spatial information in the image, and it only looks at pixel intensities. So that's actually a pretty big drawback. So just to give you a bit of an example from the images we're going to be working with, here on the left you have it's a synthetic image. I synthetically generated some pseudo receptor density images from autoradiographs that we're working with. And so if you apply Otsu thresholding here, well, you see the histogram of the image intensities. And then the algorithm finds that the optimal cut is here. And so this is the segmentation that you have as a result. So it works okay. It seems like it's a bit blotchy and blurry, but that's I guess part of the limitations. So now, if we turn to the Google Colab, where for those of you who are interested in trying out some code, we can run some stuff together. At this point, are there any questions actually so far? Oh, yeah, my slides are not in full screen, but and are there any questions? Nope. Okay, so I'm going to assume that those who want to run the code have loaded it in Google Colab. So the first thing to do is it's a Jupyter notebook, so it's a bunch of cells with code. If you are new to programming, you've never programmed, or you don't know anything about Python, that's okay. All you need to do is push the button here, the play button here, run anyway. And what it will do is first it'll pull the GitHub repository that we're using. And in there, you should see this now. If you click on this little icon here, and if you go to PNG, so this has the data that we're working with. So you can double click on it and you'll see some of the example images. Okay, so next, we go to the next cell down, which is the one with thresholding. And all you have to do is push play here. And basically what's going to happen is that we have a function defined here to sort of apply the thresholding and the segmentation. And here we have some code that's going to iterate over the file names and the images that are in the data set. And so for each of these images, we're going to apply the thresholding segmentation algorithm. And it's quite simple. All you have to do is call the threshold Otsu function with your image. And then it returns a threshold value, and then you can use that threshold value to split the image in two. So in theory, if we push play here now, it's going to generate a bunch of thresholded image, which you should be able to see in the threshold directory here. And I'll click on one. So here, there are two sets of images. There's QC images, and there are seg images. So seg is the actual segmentation. Well, it didn't work very well there. And then QC shows you the original image and then what it looks like after it's segmented. So here, for example, you see that there's some weird sort of like background artifacts here. And so as a result, the thresholding doesn't work super well. Here, on the other hand, it works quite well, although it does pick up some of this background artifact. So for those who are interested, you can play around with this code for a minute or two. There are some different things you can try. So I've talked about Otsu thresholding because that's, I think, sort of like a classic technique in this genre. But there are other ones as well, which you can try for yourself. So there's threshold_Li, which is a different function, threshold yen, threshold mean. And so if you replace this with one of these other functions, you should be able to run this cell. Make sure to run the first cell first and then this one. And then you'll get different segmentations. And you can kind of compare them and see maybe if one works better than the others. Oh, also, I should note that up here in the first cell, so here we load the images, all the images in the PNG directory. And then here I've identified a couple of images that are kind of hard, that are challenging. And so these are probably, if you don't want to look at all of the images, because there's kind of a lot of them, you can just look at these to see how well the thresholding algorithms perform. And so to do that, all you have to do is uncomment this little piece of code here. So if not file name in hard images, continue. So basically it'll skip anything that's not in the hard images, and it'll only run the other segmentation algorithm on those. So you guys can try that right now. For those who have never programmed or are a bit intimidated by Python or whatever, I'm going to go through it myself. I won't talk so I don't distract people who are fiddling with the code themselves. Yeah, so you can follow along with what I'm doing if you want to. And we'll do this for maybe, I don't know how many people are actually following along with the examples. So I'll check it in 2 or 3 minutes.
A show of hands for Thomas so that he can see how many people are trying to follow. Great, thanks. Thomas, while
Yeah.
you're doing, can you help us through the process of what you're doing exactly?
Oh, sure. I just didn't want to distract people who were fiddling, but sure. Okay, so all I'm doing here is changing the, so there's different functions for thresholding, and I'm trying different ones and seeing kind of what different effect they have. And so actually something that's kind of interesting, I think. So if we try, for example, threshold Lee here. Yeah, so it will detect the tissue. Unfortunately, it also picks up other like background stuff. So here the threshold is probably a bit too low. Although, but on the other hand, with I think Otsu, for example. Yeah, so here the threshold is too high. And so from the ones that I've tried, it seems like none of them really work well on this particular example. So again, this is one of the 3 hard images that I identified. I think part of the problem is just the fact that you have this like bottom corner of high pixel intensity, which will throw off the thresholding, of course, because then this appears like the foreground, or maybe this appears like the foreground. And so it kind of highlights the limitation of doing a intensity only approach, because you don't take into account sort of spatial features. So for example, as like humans, we can clearly see that there's like a shape here, but the thresholding doesn't know anything about that. So if you guys, can I get a show of hands if people want me to hold on this a bit longer, or if not, I can move to the next example. Okay, oh, okay, we'll hold a little bit longer. Okay, in the interest of time, I'm going to move on to the next example, which is doing the same sort of segmentation, except with k-means. So for those of you who don't know, k-means is a very widely used sort of classic clustering algorithm. And the way it basically works is that you say that you want to classify a data set into a certain number of classes. So in this example, it's 3. And then you initialize random centroids somewhere in your data set. And then in the first step, you identify which data points are closest to each centroid, and then you assign those data points to that centroid. So for example, here, this is closest to this centroid, so this gets assigned to that. These are closest to this centroid, so they become all green. In the next step, then you recalculate the mean, or you calculate the mean of the data points, and then you move the centroids to that new mean. So for example, this moves from here to on top of the only data point that's in the red category. And then you begin again, and you recalculate which data points are closest to which centroid. And then you, again, subdivide the image, and you continue like this until the algorithm stabilizes. So you can do this with 3 classes if you want. You can also do it with 2. and then you can use it for image segmentation. So there are advantages. Again, it's pretty fast. It's fairly robust. You can specify multiple classes. One problem is that it assumes a Euclidean distance between the data points and the centroids. So it kind of assumes a certain type of data distribution, which may or may not be applicable for your data set. You also have to specify in advance the number of classes you want to use. In our use case, that's fine, but in other cases, you may not know how many clusters you have in your data. And again, like the Otsu thresholding, it ignores spatial information. So if we just look at a quick example, looks about like what we saw before. So now, close this. If we return to the Google Colab, so if you go down to the next cell, the one with k-means, we can do the same thing as before. I will just run it on the so-called hard images, so it goes a bit faster. And we can push enter. Yeah, okay, so again here, it doesn't work very well on this particular image. I think if you run it on all of the data, you'll see that it actually does work pretty well on average. Oh, I only have it run for a couple. So these are all bad. Let me run it on all of them. Okay, yeah, so this is pretty good. Yeah, so you can see that it can work well, but again, on the hard images, not so much. So k-means is one clustering algorithm that you can use. However, there are a lot. Within the sklearn package for Python, there's a lot of different ones that are proposed. And so if you want to, as a quick exercise, you can try and run a different clustering technique called agglomerative clustering. Let me stop this. And so basically all you have to do to try it out is first we're going to comment out the k-means model. We're going to uncomment this line where we resize the image. The only reason we're resizing it is because you don't want to use too much RAM, too much memory when you're running the model. Because if you keep it at the 256 by 256 image size, it'll probably crash. And so then you can run agglomerative clustering, which is just another algorithm. Although you don't have to specify the number of classes, so that's kind of interesting. Also, the only parameter here that I've specified is the linkage function. So that sort of like determines how different data points in the data set are agglomerated together. I won't go into what it means, but you can try, there's also other ones you can try. I don't think I wrote them down, unfortunately. There's also single. I'll look them up quickly, and then you guys can try them for the next couple of minutes and play around with the code how you want and see if you can get these hard images properly segmented. I see there's a question. What do the warnings refer to? Oh man, okay. So it's because we're converting an int32 image to unsigned int8 with a small range. And so ideally, if you want to get rid of the error, you'd have to right, so you'd have to convert the image to uint8 before saving it, I think, or yeah, before writing it. So I see that, I don't know what this means. So if you want to try different versions of the agglomerative clustering, you can try ward, complete, average, or single. And I guess while you guys are trying to run this, I'll maybe explain a little bit more what's going on here. Actually, you know what? Yeah, for the, if we go back to the k-means briefly, just this. The first step is that you have to identify, well, so in this particular case, because we have a very good idea about where our clusters are going to be, we're going to have clusters of very low intensity pixels and clusters and a cluster of very high intensity pixels. We can define initial starting points for the centroids that we're going to use for the k-means. So in this case, it's very simple. It's just one centroid starts at 0, and then one starts at the image max. And so we define the initial centroids here, which is 0 and the maximum of the image. And then when we set up the k-means model here, you just pass the initial starting points and the number of clusters you want. So here I just take the length of the initial number of the initial starting points for the clusters. But you could change this. So what I've done in a different application, where with the same data set, I was segmenting to 3 classes, background, white matter, and gray matter. I had another starting point here, which was basically the image mean. And then you just pass that to the k-means algorithm. And in cases where it works well, it gives you one cluster closer to 0, which mostly captures the background, another cluster centered around the image mean or wherever the centroid moves, which captures the white matter, and then one centroid that captures the gray matter. So here in the sklearn sort of programming package, you define your model with whatever parameters you want. So you see it's the same basically for k-means and agglomerative clustering. You define your model, and then you can run a method called fit-predict. And so here you just apply the model to your data. And there's a bit of reshaping going on here. You can kind of ignore that, except for if you actually want to run this yourself. It is necessary to reshape it first to a vector to run fit-predict, and then back to an image. So let me try and run the agglomerative clustering and see if it actually improves the results over the k-means for the hard images. Let's try ward. Nope, no luck with that. Let's try single. Okay, that doesn't look good at all. I don't know what's going on there. Okay, yeah, no, it doesn't. So is there anybody who wants me to stay on this example a bit longer, or otherwise I'll move on? So you can give me a show of hands if you want to stay on this. Okay, there's one person. I'll wait a little bit longer. Well, yeah, there's not a lot of time left, and I still have to get to neural networks. So, I won't go full screen because well, okay, I'll try, but I'm afraid that it's going to that you might not be able to see my screen. Can you guys still see my screen? I don't hear anything, so I'm going to assume yes. So the next thing I want to talk about is watershed segmentation. And it's kind of an interesting technique because it views the image kind of like a topographical map. So where like the pixel intensities represent different heights. So bigger intensity, higher pixel intensities mean like a higher point on a map. And what you do is you basically set a couple of seed points, and you start to fill the image with water, quote unquote, from the seed points. And then you create boundaries where the different sort of like water reservoirs meet as the image fills up. So it might sound a little bit abstract, but let's say if you started with a very simple image here, like an A, where you have two black points, then you transform it so that you have sort of like a gradient. And then basically you select your seed points in the middle of these two black dots and you start to fill them, so to speak. And eventually the water the water from each reservoir will meet here, and so you define a boundary here. So then you can split the image into these two chunks. In the interest of time, I'm going to skip over the worked part of this, but you guys can go do it for yourselves. In practice, I find this technique probably works really well for some applications, but for this segmentation algorithm, for this segmentation problem, a bit less so. It would probably work better if you have, let's say, like maybe like a bunch of somewhat overlapping cells, and you want to be able to split those cells apart. That might work better. This might work well, I mean. So the last technique that I want to talk about is neural networks. And the reason why we kind of turn to neural networks is because they can be quite powerful. So you can solve really complicated problems like you see here, where you have, I think this is probably a stroke, and then you can create algorithms that can automatically segment this. And the reason why these are quite powerful is you don't have to specify a lot a priori. The algorithm is actually quite powerful and can learn a lot of different things about the image. And also there are special neural networks that are designed to take into account how spatial information is sort of laid out and correlated. So that's a big improvement on the other simpler techniques we were looking at. You can also use neural networks for very simple but annoying problems, which is what I did in some of my work. So this is the type of image I was working with, and I want to get rid of these bounding boxes automatically. So I trained a neural network to automatically recognize them so that I could then remove them automatically. So neural networks are good for both complicated things, but also simple things. As I mentioned here, though, the thing is that you need your data, so like your original images, but you also need labels. You need like a ground truth that the algorithm can learn. And so that's often one of the big limitations to using neural networks, is that you don't have access to a lot of high quality labels, especially for like medical stuff, where you need to like get it from maybe like a trained neurologist or something like that, who's very busy and doesn't have time to go through like thousands of images and carefully draw out lesions on them. So to give you guys a very brief overview of how neural networks work, I presume most of you are neuroscientists maybe, or know a little bit about how a neuron works. This will make this a bit easier. So artificial neural networks are based on artificial neurons, which work as follows. Basically, the artificial neuron receives inputs. And so this would be x1, x2, xn. And then it weights these inputs. Yeah, it weights these inputs for how important they are, so to speak. So this is kind of like the dendrites here. So this could be like the amount of X could be like the amount of neurotransmitter in the synaptic cleft, and W could be, for example, the amount of neurotransmitter receptors. And then this is the cell body, where all of the information from the dendrites gets combined together. So it's quite simple. You basically just multiply X by W. Then you transform it with this function f. So f is actually very simple in practice. It's often just returns x if x is greater than 0. Otherwise, it returns 0. So this is called a rectified linear function. And then it passes this on to the other neurons to which it is connected. So these are often organized into a network like you see here. So this is a very simple one-layer network. So you have your inputs here that pass on the input to these other artificial neurons that will sum up the inputs together and then pass them on to another layer. Often you'll have multiple hidden layers, and that's what's referred to as a deep network. So today it's possible to train networks that actually have lots and lots of layers. So what happens eventually is after all this computation is done, it passes it to the output, and then the output is compared to the label. So the label is the ground truth that you specified in advance. And that's why this is called, yeah, I guess it's supervised learning. Yeah, supervised learning, sorry. Because you're kind of supervising the training of the algorithm through the labels that you give it, the ground truth that you give it. An error is calculated between the output and the labels, and then this error is used to fine tune the weights. So to fine tune W, fine tune the weights between the artificial neurons so that it can learn better. So neural networks as such are not that useful for image processing because essentially everything is connected to everything else. And so that means that you're not really taking into account the spatial distribution of the information. That may sound a little bit abstract, and don't worry too much about it. There's something else there's a version of neural networks that are used for image processing called convolutional neural networks. And to get an idea of how those work, I first want to explain what a convolution is, if people don't know. So the idea is that when you're doing a convolution of a kernel and an image, is that you have a small kernel, kind of like a small image. In this case, this is a 3x3 kernel. And then you slide it over an image. So that would be like the big blue image here. And in each position that you slide it over, you basically take the dot product between the kernel and the image, and you get an output image. So basically for each cell or each pixel in the kernel, you multiply it by the corresponding pixel in the image for all of the pixels in the kernel, multiply them all together, sum them, and then that gives you the output here. In convolutional neural networks, it's basically you have a bunch of kernels where the values inside the kernel are basically the weights. And so what the algorithm learns is what are the optimal weights in each of the kernel filters that you're using. So that sounds very abstract, and if you didn't follow that, don't worry. I'm going to try and explain it a little bit more. So if you've never worked with like a kernel, it might seem like, what on earth are these things? But if you were to take this original image here and convolve it with this kernel here, so it's a bunch of -1s with an 8 in the middle, it basically acts as an edge detector. So this kernel is actually able to pull out a feature from the original image just based off of like the structure of the kernel. So basically it's just to say that kernels can be used to enhance an image, blur an image, or extract features from an image. Now the challenge is, like with these kernels, these are already well known in advance to have certain properties, and we don't necessarily know what kernels would be useful for looking at our brain images. What features are relevant for learning the particular task that we're interested in. So instead of using a traditional neural network, we use a convolutional neural network. Basically, the way that works is you have some kind of input image, and then you have some kernels here. So here we have three 3x3 kernels. And so for each of these, we slide it over the image, and then we make a prediction. So this would be a 3-class segmentation. We make a prediction for whether or not a pixel is gray matter, white matter, or background. And so based off like how well the prediction works compared to the ground truth, the known labels, we can then update the values that are inside the kernels to modify them. And so that over multiple iterations, the kernels will get better and better at picking out the relevant features for doing this segmentation task. So yeah, I won't go too much into this here. An important reason why these work well is, okay, so I said already that the convolutional neural network can learn features from the image, but it can also learn high-level features. And the reason for that is that at each subsequent stage in a convolutional neural network, so here you have one stage, and here would be another stage and another stage. At each subsequent stage, the kernels essentially see a bigger and bigger portion of the image, and so can extract larger and larger and more high-level features. And the idea behind this is the same as for neurons. Each of the kernels have a certain receptive field, so that means sort of that they respond to a particular portion of the image. So just to illustrate that for you a bit more graphically, so if you have this kernel here that you're sliding, well, actually, no, sorry. If if you have this point here, in your, so this would be first layer of the network, second layer, third layer. This point here is the result of a kernel that has passed over this region here. And within this region, this is the result of a kernel that has passed over this portion of the image. And this is the result of a kernel that's passed over this. So basically all that is to say is that when you're in the third layer of the image, this point, this pixel here, kind of summarizes information from a very large window in the original image. So what that means basically is as you add more layers to your network, the receptive field for the deeper layers increases and gets larger and larger. And so it can pick out or identify really high level features in your original image. So to summarize why we would be interested in using convolutional neural networks, so the filter kernels automatically learn the relevant features in the image. This is super important because it means that you don't have to specify what your kernels look like in advance. The network will hopefully just learn them. The kernels take into account spatial information in the image, which is important because you might have, for example, regions with low pixel intensities, but where you can clearly see that there's like a boundary and where something clearly like belongs to the brain, even though it's a region with low pixel intensity. And finally, deep layers can learn very high level features because they're stacked. And the receptive field for the artificial neurons increases as you go deeper into the layers. So hopefully that made sense. If not, I'd be happy to answer questions. And so finally, the network, the convolutional neural network that we're going to be playing around with in just a second is called a UNet. So you guys may be familiar with this or not, but it's a very widely used and very successful segmentation convolutional neural network. I won't go too much into the details, but basically short story is if you start with this image, which I think is like a picture of the moon or like the moon's surface, the neural network, as it advances deeper and deeper into layers, it also downsamples the image and then re-upsamples it, and while at the same time passing forward information from earlier stages. So basically it combines early stages with late stages, and this works well for reasons that we can get into. But ultimately it produces this predictive mask. So the idea here is that I think they're trying to identify lunar craters. So I think that's everything for the neural network slides. I will, oh, there's a question. When comparing clustering. Oh, I don't, so I'll answer. I don't know what that means. Okay, so I'm gonna answer that question now before going back to the worked example. I think I might have partially answered this, but I think the watershed works well in cases where you have like overlapping things and you want to be able to like split them apart. So like a common sort of worked example you'll see online is like if you have a bunch of coins that are like next to each other and they're kind of like touching. If you just use Otsu thresholding, it would basically have a foreground where you have all the coins and then a background. The watershed would probably work well for separating all of these coins apart from one another. Or again, like if you're doing very like high resolution histology or something like that, where you can see individual cells, then it could work well for that. In my experience, it seems very dependent on which seed points you pick. And if you can clearly identify what are the seed points, then you have a better chance of it working well. If you go through the worked example, you'll see that if you change the seed points a little bit, you get very different results. So for our worked example, it's not very stable. Right. So for the neural network, okay, so there is a cell here that you don't need to worry about, just run it. You can look at it if you're interested in the details of how the network is constructed, but otherwise it's not relevant. And so here we have the last cell where we actually run the neural network. And something that I mentioned earlier is that in order to use neural networks, it's a supervised learning technique. So you need to have ground truth labels. And so here, the ground truth labels that we're going to use are just the segmentations we have from k-means. So in theory, what this means is that at best, this neural network could work as well as k-means. So it's not super useful by itself, but it's okay for a worked example. And actually, you can actually use this sort of approach to sort of bootstrap some labels, which I'll explain in a second. So I'm just going to run this quickly. Oh, it's complaining. Oh, of course, I'm running into, oh, shoot. Okay, so the problem, I'm going to rerun this with, I need to rerun the k-means because I think I didn't write all of the images that I needed, or they might have not been resized correctly. I'm not sure. But in any case, I'll rerun k-means quickly, and then we can run that.
While you're doing this, Thomas, there's another question in the Q&A.
Okay. 1 second. Voxel size. I mean, I guess you don't. Let me see if I understand the question. I need a little bit more information as to what you mean by account for the voxel size. I mean, I guess, do you mean that if like the voxel isn't isotropic, I mean, I guess essentially you don't. If it's relevant, it'll probably be learned by the network in some way, but yeah, I mean, I guess it could get weird if you have like really extremely non-isotropic voxels. Yeah, but even so, you would have to account for that, I think, in the pre-processing of your data, where if you think that that's a problem, you'd have to resample it. I think that's what I would say. Oh, shoot. Sorry, I have to rerun it on all the data, not just the... Okay. So while k-means is running, I'll explain what I meant by bootstrapping some labels. So what you can do is, if you need to create a bunch of labels and you don't have a lot of time, you can use something like Otsu thresholding or k-means first, run the network on that. And so, of course, it's going to be as good as your initial segmentation. But then it may fail in maybe like a handful of the sections, so maybe like 10% or 20%. And then you can go back over those 10, 20% manually and like correct them. So instead of going through like all 200 or all 1,000 sections, you only go through 100 or 200. And then you'll have much better labels, and then you can retrain your network on the new set of labels, and it'll hopefully work better. And maybe you only have to correct like a 5% or 10% after that. So you can kind of yeah, bootstrap your way to having more labels in certain cases. Okay, is this still running? I think this is done. Okay, it looks like it's working. All right. So here, the first thing that it prints out is sort of like a summary of the of what the network looks like. So yeah, you have a bunch of layers here. I won't go through it. Just know that if you're doing this yourself for your own application, one of the hard parts about neural networks is not actually like the underlying math or whatever. Like the packages that are available are very good at taking care of that for you. What's hard is having correctly formatted images that are the correct size. So I would highly recommend making sure that all of this makes sense on your own data when you run it. So here we just trained for one epoch. So we basically just did one training set on the data, which is not enough, but it gives us some output that I can explain. So there are basically two stages in in an epoch. First, it trains the model, and then it gives you a loss and a sort of accuracy metric for the training. So you see that here. Loss is the actual error that the network is using to improve over time. The metric you see here, categorical accuracy is like a more human readable assessment of how well the model is performing. So ideally we would want this to be at 1. So at the moment that's 6.62. And here we have the validation loss and metric. So basically after the training, the model will be applied to a certain subset of the data that is reserved exclusively for validation. And so here it gives you the validation loss and the categorical validation accuracy. So the fact that this is much lower than this is a little bit worrying, because basically what that means is that the network has maybe overlearned on the data that it has, and so actually doesn't generalize very well to new data. However, I think in theory, last time I ran this about 2 hours ago, it should improve over time. So if you were to run this for a few more epochs, it should work better. And so we see here what happens when you just learn for one iteration. I mean, you have, it looks like the kernel, the network has learned some kind of like edge detection. So yeah, one epoch is definitely not enough. But with the remaining time we have, you guys can try and tinker around a little bit. Unfortunately, we don't have a lot of time left, but you can definitely like try to put this to 10 epochs if you want, and then see what happens then. You can also tinker around by changing the network itself. So if you go in here, these convolution 2D layers, so this is the layers that specify the convolution kernels that you use for each layer. So here the first number is how many kernels there are in that layer, and this specifies the size of them, so 3 by 3. But you could change this to 5 by 5 or 7 by 7 if you wanted to. You could change this to some other number. So you guys can start playing around now, and I'll tinker around a little bit on my own, kind of explain what's going on for people who are not interested or not who aren't tinkering. Yeah, okay, so here, this is actually what I was expecting a little bit more. So it starts out very low, so the categorical accuracy on the first epoch is quite bad. But it seems to be increasing over time. So again, this is the training metric. So the first one it gives you is for training. Then the second one is for validation. The validation is going up a bit as well, not as quickly as the, not as much as the training validation. Okay, so yeah, so it seems to be learning pretty well. So we'll see how well it performs. When you're running this on yourself, something to make sure is that the training loss and validation loss, that these are going down, because the metrics are not exactly what the network is training on, it's on the loss. And so if the loss is going down, it means that it is in theory learning something. Because sometimes if you have like a poorly specified model or crazy data or something like that, like your loss might do crazy things, it might jump up, it might jump down. And so that would be a sign that there's something really wrong with the way you've either structured your data or your network or something like that. So right here, we see that the validation accuracy has started at 0.25 and has jumped up to 0.69. So that's not too bad. If you start working with these things, you'll end up spending a long time kind of like staring at the screen as these numbers kind of hopefully go up, like you're watching some kind of like sports match or something like that. Which is maybe not the best use of time. Okay, so here the validation accuracy is higher than the training accuracy, which is actually a good sign. It means that the model still has sort of room to learn. If your categorical accuracy starts getting really high, but your validation accuracy stays stable or goes down, it basically means that you're overlearning, you're overfitting the data. And so that's really bad. That's actually one of the things that you want to avoid. Also, something to note is that in the actual code, here. This is a bit arcane for anybody who doesn't program, but basically we're not including the _B images. So here you see that all the images have a label associated with them. And so the model isn't training or validating on the _B ones. And so those are completely new to the model when at the end, the model gets applied to all the images. So afterwards, what you can do is go check the results on those B images. And those are images that the model has never seen before. And so it shows you to a certain extent that this sort of neural network can generalize to completely new data or to relatively new data. And so I think that's kind of like a neat feature of this. Okay, so we're still going up. That's good. So I think now it's finished training and it's applying the model to the images. So for all the input images, it's trying to segment them using the network. While that's going on, something that I added to the network here are dropout layers. So what these do is they randomly knock out a certain portion of the weights at each iteration. So here, because it's 0.1, it's going to knock out 10% of the weights at each iteration for the previous layer. And so what that kind of does is it prevents, it helps regulate the model and prevent it from over learning and over depending on a certain, on a small subset of weights. So what could happen is that you could have a handful of artificial neurons and weights between them that happen to just randomly be really good at predicting your training set, but may not generalize well. And so by randomly knocking out 10% of your weights, it prevents the model from kind of like finding this suboptimal solution. Okay, so neural network. Oh, so it doesn't, that's weird. Huh. So it doesn't seem to be performing very well. I wonder why that is. So when I tried it before, it did actually segment well, but I guess here it's not. I'm not too sure why. Maybe, so here we see that the categorical accuracy is at 0.74 and here it's at 0.77. So we could potentially train for longer or add more, more filters at each layer. But yeah, I would have expected it to work a bit better. I mean, in any case, that's sort of how it goes with neural networks. You got to experiment and tinker around until you find something that works. Although make sure you don't overfit your data. That's definitely a serious problem. Before we, oh, I have only a minute left. So, oh, there's 2 questions. So will the network be better than k-means? Yeah. So because k-means will like pick up artifacts or things that shouldn't be in your segmentation, if you get rid of those manually and then you retrain the model, then it will be better because it'll have learned to ignore those things that k-means would otherwise pick up incorrectly. And an interesting thing is that actually neural networks can do better than the segmentation that they're trained on. So for example, let's say you were to do like a cortical segmentation with FreeSurfer. What I've seen before is that if you train your network to learn the FreeSurfer segmentation, the network will actually work better, or can work better. Because the FreeSurfer has little bits of like variability. So maybe sometimes like 2 sulci will be fused together when they shouldn't be, and sometimes it might miss a little piece of cortex. And so all these little pieces of variability will sort of, the network will learn enough to be able to sort of like spot those mistakes and won't necessarily make those mistakes itself. And so actually you can get a bit better performance from a neural network that's trained on a segmentation than you just get from the segmentation. I'm not sure if that would be the case with k-means, probably not, but yeah. I see that there's an open question. Oh, okay, I see Claude's question. I think I mean, depends on the 3x3 stencil is maybe better for it depends on the features the size of the features that you want to identify, and also the number of layers that you use. Because even if you just use a very small stencil, like a 3x3 stencil at 10 microns, if you have a bunch of layers, let's say you have like 20 of them, by the time you get to the 20th layer, it might have a field of view of like 5 millimeters or something like that. So it really depends on how you structure your network. Also, you can use dilations, and you can change the stride, which are different things to change like the receptive field of your kernel. So I think I'm out of time. I'm over time. So thank you very much. If you have any questions, you can email me, and I'd be happy to answer them.